Now let's take a look at our own study. To begin with, we gathered 10 datasets from both Pandomics and Gene Expression Omnibus. Here's a screenshot of these 10 datasets, and since we didn't find enough that satisfied our requirements on Pandomics, we asked the Omics team to upload some of the new ones we found on Geo. And so many thanks for their quick help and effort. The experimental design was cases against controls. Each study either had separate control patients or a healthy tissue sample and a diseased tissue sample from the same patient. Here's a heat map which demonstrates the analysis of correlations between our datasets in the glioblastoma project, and each square indicates the correlation in the total change in global gene expression profile between two compared datasets. What's amazing is that we took entirely independent datasets from different platforms made by different authors who had different aims and even were published at various times and still got a strong correlation between each of the datasets. The dark red color, which shows that the profile of expression of all proteins in our dataset is very, very close, could signify that the characteristics of this disease is preserved despite all of the factors which could have caused this correlation. So we looked through all of our individual comparisons in the different datasets between healthy and ill patients in order to understand which genes and which cell processes were the most strongly affected. After a good look, we chose to pay special attention to these cell processes, which you can see are indicated in red on the slide. All our datasets showed very similar results for these pathways, as you will see very soon. The first three pathways are connected to DNA repair, DNA replication, and expression, and these are all upregulated in our datasets. The fourth pathway is the neuronal system, and you will notice that it is downregulated everywhere. The fifth one is connected to the immune system, mostly upregulated, and the last few pathways are connected to programmed cell death and the cell cycle and chromatin organization, and these are all upregulated, while vehicle-mediated transport is mostly downregulated. Now, if we look at all our other datasets, we see the exact same picture. Then we selected one specific dataset for further review. It was GSE 108474, and the reason we chose this is because it had the most cases and controls out of all of our datasets, and it's also a relatively new study made in 2017. So let's take a look at its differentially expressed genes and pathways. The four pathways chosen were CD163 mediating an anti-inflammatory response, the transfer of LPS from LBP carrier to CD14, neuropilin interactions with VEGF and VEGFR, and the AIM2 inflammasome. I'll be taking you to Pandomics for each of these pathways and demonstrate how and why we chose them. So let's begin with CD163 mediating an anti-inflammatory response. The first thing we did was order the pathways from highest to lowest high panda activation. And above are listed cell processes, which Pandomics colors in a very specific way that lets us see what is up and downregulated in glioblastoma patients. So let's rank everything by IPAND activation. We see our chosen pathway is third on the list, and this already indicates its important role in glioblastoma development. Literature on this pathway is overall very new, so we're motivated to look at exactly this pathway. Some publications even show different gene expression in varying subtypes of glioblastoma, like proneural, neural, mesenchymal, and classical. The function of this pathway is mainly homeostatic. However, it also takes part in adhesion to endothelial cells, tolerance to induction, and tissue regeneration. CD163 loss inhibits cell cycle progression and proliferation of glioblastoma, as the study showed. And the same investigation noted that knocking down CD163 impaired glial stem cell stemness. We agree that this pathway may serve as a therapeutic target for glioblastoma. On the photo, bottom left, you can see the pathway's mechanism of action. CD163 causes M1-like glia to become M2-like glia, which causes anti-inflammatory responses and interleukin 10 and 4 release. And such action will make it more difficult for the immune system to attack the cancerous cells, concealing them thereby longer. On the right photo, you can see the subtypes of glioblastoma, classical, mesenchymal, neural, and proneural, and CD163 is most strongly expressed in the mesenchymal and proneural glioblastoma. Perhaps this data could be used to target CD163 and specifically these two subtypes of glioblastoma if creating drugs against this pathway. Now, with the help of Pandomics, we created box plots showing CD163 expression for each of our datasets. All the box plots are made on a logarithmic scale, and for this gene and for the ones that we will show in the rest of the presentation as well, many of them showed very high expression on this gene and in comparison to patients without glioblastoma. Now let's look at the neuropilin interactions with VEGF and VEGFR. The first thing we did was order the pathways from highest to lowest IPAND activation. This time, take a look at signal transduction. This is an important process in cancer proliferation. And here is our pathway in the fourth spot. So you might wonder, why didn't we choose one of the first three? Well, we know that VEGF is a strong biomarker of glioblastoma from reading literature and learning about studies, but what makes the fourth pathway different is that there are neuropilins involved. There hasn't been that much data on neuropilins, and so we decided to investigate specifically this pathway. It has 4 different genes involved, 2 neuropilin encoding ones and 2 kinase coding ones, and all of them are upregulated. Neuropilin 1, VEGF, and VEGFR receptors have shown to promote glioblastoma development. In fact, glioblastoma cells from patients that expressed these genes attenuated cancer stem cell markers, which led to higher migration rates of the tumor. Studies have also proposed blocking the VEGFR2 kinase, as this would stop the neuropilin pathway and could help treat glioblastoma. On the image on the right, you can see how the cancer grew without any inhibition, and then with NRP1 and VEGF inhibition in mice xenografts. You can see from the smaller slope of the line in figure D that the tumor size was much smaller in mice with VEGF or NRP1 being inhibited. And figure E shows that with the inhibition of VEGF and NRP, mice also lived longer. And I want to draw your attention to the fact that bevacizumab, a drug used against glioblastoma, is used to target the VEGF pathway. We found, however, some investigations that showed inhibiting NRP1 yielded better results than inhibiting VEGF, so maybe this could be another direction to look into in order to find better therapeutic agents. We also made box plots for this pathway on a logarithmic scale as last time, and from the slide you can clearly see that glioblastoma patients have higher NRP1 expression. And the same with NRP2. Glioblastoma patients have this gene upregulated as well. Now let's look at the next pathway. To further research in possible immunotherapy against glioblastoma, we wanted to check out immune system cell processes. The very first pathway is transfer of LPS from LBP carrier to CD14. Good, it has a high IPAND activation and a low p-value, so we choose to look at exactly this pathway. The 2 genes in this pathway are CD14 and LBP. And CD14 is a monocyte differentiation antigen, while LPS is a binding protein. CD14 has 2 isoforms, one of which is secreted in the extracellular compartment, while the other is anchored to the plasma membrane via its GPI module. This gene is expressed and released from human glioma cells and infiltrates macrophages, which may be observed as immunodepletion in patients. In other words, this pathway is related to concealing cancer cells from immune cells. The photograph shows the way that the LBP molecule and opsonin and SD14, an opsonic receptor, work. LBP binds to the TLR4 receptor, and CD14 connects to the complex, which causes a cascade leading to an effect on NFKB and AP1 transcription factors, and these two mediate a lot of different gene expression. We created box plots on a logarithmic scale for this pathway as well, and CD14 was upregulated in 7 out of 10 of our datasets, showing that glioblastoma patients have on average a higher expression of this gene as compared to healthy controls. And now the final pathway is the AIM2 inflammasome. So you can see that the IPAND activation is quite high for this pathway, and it has a low p-value as well. And we didn't choose the three above as they were all quite well researched in regards to glioblastoma, while inflammasomes are a frontier topic and it doesn't have as many publications about it. The inflammasomes form the central hub of innate immune regulation. AIM2 is a member of the nucleotide binding domain containing the NLR family of proteins which assemble into multi-protein complexes when under pathogenic assault. And this inflammasome assembly leads to the secretion of bioactive interleukin 1 beta and interleukin 18 and to the induction of pyroptosis. The addition of dsDNA to double-stranded DNA to cultured neurons enhanced axon extension and downregulated dendritic growth, leading to the regulation of neuronal morphology. Studies have shown that AIM2 expression went up in glioblastoma patients as compared to healthy patients. On the image on the right, you will see the AIM2 inflammasome pathway in more detail. And we also made box plots in pandomics for this pathway and saw large differences between healthy and diseased patients. You too can see here that the AIM2 expression is higher in glioblastoma patients.